Hey there, biologists! In this set of notes, we are going to be learning about the history behind how DNA was discovered to be the hereditary molecule or material of all life. This set of notes corresponds with this notes template where I will give you the who was involved, so the names of the scientists, where the research was conducted, the time frame, the purpose. I've given you the hypothesis for each of the experiments, and I've started filling in the experimental design for you, but you will want to finish filling that in. And then you'll also be putting in what were the results of that experiment, what was the conclusion of that experiment, and why was that significant, or what was the importance of it. This video will also be intermittently interrupted with drawing notes where you will label the specific parts of the experiment to help you better understand it. This is absolutely critical because these experiments are kind of complex, and without taking the time to watch the video and illustrate your notes, I think you'll really struggle to understand the concepts. So make sure that you're utilizing these notes as efficiently as possible. We've also broken down the set of notes into two halves. So the first part of the notes will be the Avery experiment and the Griffith's transformation principle experiment. And the second part, which you'll do the following day, is the Hershey-Chase experiment. Okay, so let's go ahead and get started. So scientists, on their quest to understand what the genetic material of all life is, they posited or thought that there are three requirements that genetic material must meet. Can you guess at least one of what these three requirements would be? Think about what you've learned so far about cells and cell division, or the function of cells. All right, well, let's go through what they realized. So scientists reasoned that there are three requirements that genetic material must meet. Whatever the genetic molecule is, it must be able to store information that can determine the development, structure, and metabolism of a cell. They also posited that it needs to be stable so that molecule can be replicated for the process of cell division. And lastly, they reasoned that the molecule must be able to undergo changes over time, because as changes occur, which are called mutations, that essentially allows for evolution and survival in different conditions. So at first, scientists thought that proteins were the genetic material. Here's why. Well, proteins are far more complex than DNA is. Take a look at this protein molecule. This is one example. You can see all the different types of atoms that form all of these complex interactions between different parts of this molecule. Whereas DNA in comparison is kind of simple. And I will pose here that they don't quite know the structure yet at this point, but they have an idea of just how simple this molecule is relative to proteins. They also realized that proteins are made of 20 different building blocks called amino acids, whereas DNA is only made of 4. And so they thought that, well, DNA is pretty simple structure, simple building blocks. So, DNA or our genetic information is really complex, so therefore proteins are more likely to be the case. They also reasoned that, well, proteins are more numerous. There are more proteins in a cell than any other type of molecule, so maybe that has to do with the fact that maybe they store the genetic information that's used to build that cell and make it work. And lastly, there's also a wide diversity. There's more different types of proteins than any other type, and maybe that can essentially contribute to the different traits that we have. So we're going to go way back in time to the 1860s, where we're going to look back at the history of the discovery of the nuclein, which is the material inside of a nucleus. This was understood or first developed by a scientist by the name of Friedrich Miescher. He was talked about in the documentary you watched. He was a biologist conducting research, and he was looking to figure out what are the different parts of cells. He wanted to isolate the molecules that make up cells, and he was specifically using white blood cells as his source. And the reason why is because at that time there was a war taking place, and there were a lot of injured soldiers coming back from war who had really serious injuries, and these injuries produced a lot of pus. And pus is made up of your dead white blood cells that have died during the battle of defending your immune system. And so he took these white blood cells from injured soldiers at a hospital, so they were in plenty at the time. He was able to isolate the material that was found in the dense nucleus through a series of steps, and he called this isolated information nuclein because it was in the nucleus of the cell. He analyzed it and discovered that what was really unique about the DNA molecule in comparison to proteins, for example, is that DNA contains large amounts of phosphorus. So DNA is very unique because of the phosphorus element, which is not found in proteins, carbohydrates, or lipids, for example. So he was able to demonstrate that nuclein was found in the nucleus of every cell of every organism that he tested. But what's tragic here is that for nearly a century, his research was not recognized for its importance because, again, most scientists at that time in the 1860s thought that proteins were the genetic material that controlled our traits. But there is one problem with this idea that proteins are the genetic material. I want you to think about what we've learned about proteins. For example, enzymes. What do you think might be one argument against proteins being a genetic material based on what we've learned about enzymes? Hint, think about what might happen if the environment changes. Well, hopefully you remember that enzymes break down or denature when they're exposed to heat. So if the genetic molecule of our cells degraded every time we get overheated, that would be disastrous. Our genetic material is really critical for our cells to be able to work. So it just doesn't really make sense. So at this point, scientists are on a quest. They want to figure out what molecule does store that genetic information that is passed from generation to generation. And so the results of the upcoming 3 foundational studies provides the evidence that is needed to confirm that the theory of DNA as the heritable material of all living organisms. All right, but before we start with the first experiment, here's a little bit of background context. So what's super cool about bacteria, which are going to be a cell type that will be used for research coming up, these bacteria can take up foreign molecules floating around in their environment. So bacteria are this single-celled organism. They have a single chromosome, and they can pick up molecules of DNA floating around in their environment. And again, just to pause, Griffith did not know that they can take up DNA. He just is going into this using bacteria without recognizing this, but I wanted to give you this background at least. Okay, so none of this was known in 1928 when Griffith is doing his experiment with bacteria and mice. So Frederick Griffith was, again, studying. You learned about Frederick Griffith briefly in the documentary you watched. He was doing his research shortly after World War I, and this is because pneumonia was a significant cause of death. It's a respiratory illness that affects your lungs. And so he wanted to try to develop a vaccine to try to prevent or reduce the amount of deaths associated with pneumonia. And so in his research, he was using 2 strains of Streptococcus pneumoniae bacteria. Again, he was doing his research in 1928, and this was in Britain. So he started with one type of bacteria called the type S strain. The S stands for smooth. You should be writing this on your notes, by the way. So you can skip the experimental design for now. We are working on, we're right here. Okay, so make sure you're adding it to the drawing notes right here. All right, so type S strain. Smooth bacteria, and it's called S strain or smooth because these bacteria, which are red, are covered by a protective capsule, which is yellow. And this capsule on the outside of this bacteria prevents your immune system from eating up this bacterium and breaking it down. So these bacteria do not get broken down by the immune system, and therefore they cause disease, and they can cause death. All right, and he was also experimenting with a second strain of Streptococcus pneumoniae. And this second strain is called the R strain, or rough. And this is because this strain does not have that outer capsule. We're just using blue dots to represent this, but if we were to zoom in with a microscope, you would actually see that there's kind of a rough texture here on the outside. And because these bacteria do not have an outer capsule, your immune system can easily recognize these as foreign and then can attack them and kill them. So these bacteria die before being able to cause disease by your immune system. So they're non-deadly. All right, so let's take a look at his experiment. At this point, we're going to transition to the drawing notes. So go ahead and make sure you have a couple different colors that you're ready for the drawing notes if you're using your iPad. So you should already have the type S and type R strain labeled on this page of your notes. Again, S stands for smooth. These are deadly or lethal bacteria. And the type R strain, R stands for rough, and they are non-deadly or harmless. Let's briefly or quickly illustrate what these cells look like again. So the smooth bacteria we're going to use red for, and they are not, or they're deadly because they have an outer protective coat that prevents your immune system from being able to destroy them. So that's why these can easily infect your cells and cause disease and in many cases death. In comparison, the rough bacteria do not have that outer capsule. So if we were to look at them under the microscope, they would have kind of a rough texture. It's hard to illustrate that, so I'm just going to draw them as blue circles or dots without an outer protective coat. So in the Frederick Griffith experiment, he, like you saw in the documentary from Tuesday, what was really cool is that he was starting to see, well, first of all, which of these strains of pneumonia causing bacteria are deadly versus non-deadly, and what happens when you heat treat those bacteria, and then what happens if you mix them together. So in his experiment, he started with the smooth, or the smooth strain, the S strain. And when he injected mice with these bacteria, make sure you're drawing this along with me. When he injected mice with this bacteria, the results of this were death. That was to be expected. That's what he predicted would happen. When he injected the mice with the R strain bacteria, these lived. Okay, because we know that those are non-deadly bacteria. When he applied heat to the S strain bacteria, similar to when you cook your food, cooking or applying heat kills bacteria. And so when you heat treat these bacteria, it degrades the cell, or at least makes them not able to infect your cell. So I'm going to do that, and then I'm going to draw like a X to kind of show that we heat treated them or killed them. And as a result of that, these mice lived. Now, what was really fascinating is when he took this living R strain, non-deadly strain, and mixed them with the heat-treated S strain. Again, these by themselves are not deadly. What's interesting is when you mix those together in a culture, so I'm going to draw both of them in this culture. What was fascinating is when you mix them together, even though both of them on their own are non-deadly, it caused death. So Frederick Griffith was puzzled. Something must be transforming these bacteria. So when we think about it, something must be transforming. So here's the living bacteria. Something from this dead S strain, something must be transforming that bacteria. But what is it? What from that deadly strain of bacteria is being inserted into the harmless blue strain of bacteria? So his conclusion is that something, here's the conclusion, something is transforming the harmless strain of bacteria into a deadly strain. Some kind of molecule right here is going from one cell to the other. But again, his focus in his research was on vaccines. His was not to solve the mystery of what molecule in a cell can transform different types of cells. So the significance here is that he could not determine what specific substance was causing this transformation, but he was able to at least identify that something is happening here, which is what the next scientists will play on in their experiments. All right, so at this point you should have completed the drawing notes, and if you need to fill in the parts that go with this, here is to go back, so you can pause here to fill in this part to these boxes here if you need. And then you can also, I'll reveal this so that you can see the experiment, which you will fill in here as the basic steps. You can keep it pretty simple, keep it sweet. So pause here if you need to fill that in. All right, let's take a look at his conclusion. So you can write this in for your conclusion right here. I think the results are here too. So Griffith concluded that something must be transforming the usually harmless rough strain of bacteria into the deadly smooth strain, but he couldn't determine what that was. Okay, so there's some kind of mystery substance, some kind of mystery molecule that's transforming these non-deadly strain into deadly strains when they're exposed to this type of molecule, or cell. All right. Experiment 2. Let's talk about Avery, McCarty, and MacLeod. So Avery, McCarty, and MacLeod built upon Frederick Griffith's experiment. They wanted to answer the question that was left unanswered by Frederick Griffith's experiment. Like, what is that mystery molecule that can transform non-deadly bacteria into deadly strains? Their research was done in 1944. So again, on your notes here, you're going to fill in the, let's see, we're on who, you'll fill in the when, purpose, that's all here. So in 1944, Oswald Avery, Macklin McCarty, and Colin MacLeod sought to determine what substance, what was that molecule that caused that bacterial transformation that was observed in Griffith's experiment. So here's the purpose. They want to figure out what is that mystery molecule that can cause bacterial transformation. They're doing the research in New York, USA. And in comparison to most of the scientific community at that time, this team of scientists actually did predict that DNA was that genetic molecule. So they're using this experiment to confirm or refute that hypothesis. We are going to transition to the drawing notes at this point. When you come back to this after the drawing notes, you can then pause and add this in for your other areas of your notes. In the Avery, McCarty, and MacLeod experiment, they sought to determine, in the Frederick Griffith experiment, what was the specific molecule that was causing the cells to transform. They decided to use the process of elimination. Similar to the documentary, you could think of cells as like bags full of molecules. We know that cells contain proteins, lipids, carbohydrates, DNA, and RNA. In this case, Avery's team determined that carbohydrates were definitely not a substance that they needed to examine. They removed that from the equation. For the following, they wanted to analyze between RNA, DNA, and protein. If they add enzymes to destroy a specific substance at a time, they can essentially determine what specifically causes that transformation. So the idea here is if they remove lipids and sugars from a solution of heat-killed S cells, the proteins, RNA, DNA remain, so that's what's contained in all of these test tubes that they have. And here's the big idea, so right along with me. The reasoning that they had is if the genetic material is destroyed, then transformation cannot occur. If the genetic material is destroyed, therefore transformation cannot occur. So essentially, they are going to use the process of elimination and multiple trials in this experiment. Let's start with the review of the bacteria that they're using in this experiment. So in the Frederick Griffith experiment, he established that if you take a deadly form of the bacteria called S form or S strain, this is the strain that had the protective outer capsule. And if you heat treat those bacteria, so here's that outer capsule, if you heat treat that bacteria, it kills them. And therefore, you turn a deadly strain into a non-deadly strain. So these bacteria that we're going to be studying are the heat-killed deadly form. So they are by themselves not deadly. So in this original batch, you have a mixture of heat-killed S cells. You have proteins, RNA, and DNA all floating around. What they then do is they create 3 different trials. In this first trial, we are going to destroy protein. So let's write that down. In this first trial, we are going to add an enzyme called proteinase, which is going to destroy protein. In the second trial, we are going to add an enzyme called RNase. And this RNase enzyme will destroy the molecule that is called RNA. It's a relative of DNA, but it is not the same thing. And in the last trial, they are going to add an enzyme called DNase, which will destroy DNA. Let's see what the final results are. So in this first trial, when they added the proteinase to destroy the protein, and then they added the harmless living R cells, and they mixed them all together, transformation still occurred and the deadly S cells appeared. Let's write that down. Deadly S appear. You can see that shown by those bacteria right here. So we've got this deadly strain. It still appeared, okay? So clearly, if protein is destroyed, the transformation still occurs, so therefore protein cannot have been the genetic material. Let's take a look at trial 2. They added RNase and they destroyed the RNA. When they added the living, harmless R cells to this mixture, guess what? Transformation still occurred. You can see that right there. These bacterial cells still appeared. So deadly S cells appear. Transformation still occurred. So therefore the genetic material is still present. Therefore RNA is not the genetic material. In the last trial, they destroyed the DNA molecules inside of those bacterial cells. And when they added the living R cells, which were harmless, in the mixture, guess what did not appear? There was no transformation. There were no S cells that appeared. Okay, so let's write this down. No S appear. Okay, so this is important right here. So if we destroyed the DNA, transformation could not happen. Therefore, we must have destroyed that genetic molecule. If we destroy the genetic molecule, it can't be exchanged into a different cell. So therefore, our conclusion of this experiment could be that transformation of bacterial cells required DNA. Therefore, DNA is the genetic molecule of bacteria. So what was the significance of the Avery, McCarty, McLeod experiment? Avery, McCarty, and McLeod's work was significant because it supported the hypothesis that DNA is the heritable material of at least bacterial cells. So they confirmed that different types of bacteria all use DNA as their genetic information. But many scientists still argued that DNA was not complex enough to be the genetic material of all types of cells, including ours. So the next set of scientists sought to answer that question. They wanted to determine what is that molecule that is used in mammals or complex organisms like eukaryotes and not just bacteria. So that's the next set of experiments that will be in part 2 of this video. So you can stop the video here for day 1.